Hello and welcome. Today we're going to be talking about prokaryotic gene regulation. So in particular, how do bacteria cells actually control which genes they express and when they express them? So for bacteria, just like all living things, metabolism is of utmost importance. They want to make sure that they can very quickly respond to their environment with any changes that might occur. And especially since that one cell is surrounded completely by its environment, it is important that it can respond quickly to that environment. Bacteria cells also want to make sure that they're not wasteful. So if they have enough of the product that they're making, they absolutely need to stop that production. Not only just time, but energy, materials, they do not want to waste. So how they're going to do that is they're actually just going to control them. Their enzymes. Their enzymes are going to make it responsible for their metabolism to happen. So if they can control when their enzymes are manufactured and how quickly they are manufactured, they can then control their metabolism. So it is going to stop their production of the enzymes then that they would have. But on the other hand, let's say they come across a new energy source that's a new food in their environment, they were going to want to use it. It gives them that advantage over the rest of them if they can make an enzyme to respond to that. So they would need this energy for growth, for healing, for reproduction. And how, again, they're going to do that is they're going to use their enzymes. So they would start the production of an enzyme, and they would want to make sure then they could digest that. So two differences here. If they are needing a product, then they're going to go through the synthesis reaction. But if they find something that they need to break down, then it's the digestion reaction. So one way that they could possibly do this is through feedback inhibition. So the end product of that enzyme pathway could possibly act as an allostatic inhibitor. That product then would change this beginning enzyme so it could not function. So it could change then the active site of the enzyme. But it's extremely wasteful to do that this way. So they have a different way in which they're going to regulate their metabolism. And they do so by not just allosterically inhibiting an enzyme of theirs, but they can actually control when genes are made. So they're going to control their gene regulation. So for this example here in the pathway, if we're going to produce tryptophan, it would be much more advantageous for this bacteria cell to not have to make all of these enzymes and waste time and resources, where they could just not transcribe a gene and have the same effect. So for bacteria cells, it can vary in the amount of specific enzymes that are regulated by gene transcription, and they're going to come in two forms, a turning off or turning on, and they can also add complexity to that. So I like to think of it kind of like a light switch where it turns it off and on primarily, but you might have a dimmer so it can also slow down or speed up as well through different transcription factors. So if the genes are turned off, then they maybe have enough tryptophan in the example on the previous slide, and they would just shut down the transcription of that gene. But maybe they don't have enough. So then they could turn on the cluster of genes. So for example, if we're thinking about metabolism and something you would need to digest for energy, maybe there's a new sugar like lactose in their environment that they would want to be able to turn on their genes. So what bacteria cells do is they group all of these genes that are responsible for coding for enzymes for either synthesis or for digestion in operons. And operons all have the same basic parts, although the number of genes that they might have at the end can differ between these operons. But all of the genes that they do have encoded are going to be for a related function, for synthesis or of digestion of a particular compound. So within the operon, you have a promoter. The promoter should sound familiar to you. The promoter then is going to be the spot in which RNA polymerase is going to attach. So it's the binding site for that. And then that RNA polymerase would be able to transcribe all of the genes that are in the operon. The other thing that they would have is they have an operator. The operator is going to act kind of like the on-off switch. So the operator is that binding site then for the repressor protein. So that repressor protein, when activated, would attach to the operator, or I should say when it has the correct shape, it attaches to the activator. Also remember that if this is a prokaryotic cell, then not only is transcription happening, and this is one single mRNA that's going to be made, but translation would also be happening simultaneously. So how can you turn these off? Well, with the repressor protein. So the repressor protein, when attached to the operator, is not going to allow for RNA polymerase to transcribe those genes. So if you have the repressor protein attached to the operator, it is not going to allow for transcription. And in the operon model, again, you are going to have the promoter. In this case, it's the Tata box, a common promoter. You are going to have the operator, and the operator can have that repressor protein attached to that. And then you have your series of genes. So for this one, you do not have the repressor protein attached at the operator, and you can see that RNA polymerase is able to transcribe the mRNA, and again, simultaneously translating that into the series of enzymes. But if you do attach the repressor at the operator, then you are not going to allow for transcription to occur, and you are not going to be generating those enzymes. So two main categories of operons. First one is the repressible operon. The repressible operon is going to be used in a synthesis pathway. So it's going to be used to create some type of compound that the cell is going to need. So in this case, the tryptophan is going to be the repressible, controlled by a repressible operon. So if I think of repressible, I think of like it can be turned off. So it could be repressed. So if it's a repressible operon, then in its normal state, it's going to be on. Bacteria cells are going to need the amino acid of tryptophan. So they're going to continually have that expressed. But so we'll see the expression, RNA polymerase not blocked is able to transcribe the mRNA, which makes the enzymes responsible for making tryptophan. But if we have a lot of tryptophan in the environment within the cell, we do not need to continue to make it. That would be wasteful. So what the tryptophan will do is it will allosterically bind to the repressor. It's not going to bind onto the active site where it would attach onto the operator, but that has a conformational change, and that is going to allow for that repressor to now attach to the operator. And that blocks transcription. RNA polymerase cannot get through. So what happens when tryptophan is present? When it is present, and it is present at high levels, you do not need to make those tryptophan building enzymes. So therefore, it's going to attach to the repressor. The repressor is going to become activated through a conformational change, and it will stop transcription. The other type of operon are inducible operons. Inducible operons, in this case, I like to think of that they could be turned on in the environment if the environment calls for it, within the cell if the environment calls for it. So oftentimes too, inducible are going to be for a digestive pathway. So this is going to be those catabolic pathways. So a really good example is the lac operon. Lac operon was one of the first examples of operons that we were able to understand. So in this case here, it should look a little bit different. We still have our promoter, the Tata box, same one. But in this case here, our operator has the repressor attached to it. We don't have anything that is attached to the repressor besides the operator. So in its natural state, without anything that has allosterically bound to this, it's going to be in this on position, and it's going to be blocking transcription. So RNA polymerase cannot attach. But if the environment changes and if we suddenly have lactose, it's going to be beneficial for the bacteria cells to be able to break that down. So in this case, lactose will allosterically bind to the repressor, causes a conformational change, and it is then going to allow for transcription and translation to happen. So what would happen when lactose is not present? Well, if it is not present, then you don't need to make those lactose digesting enzymes. So what would happen here then, instead of this model, your repressor would be attached into this inducible operon. So speaking of the lac operon, Jacob and Minoid were the first to actually hypothesize that this is how bacteria cells controlled their metabolism. So they were able to coin this term of operon. There's this operating system for how bacteria cells could respond to their environment. Hopefully, you can summarize what is a repressible and inducible operon. What I would like for you to do is in Schoology, there is an assignment where there are 4 different examples, and you need to then figure out if they're repressible or inducible. You also need to figure out if they're catabolic or anabolic, and if it is on or off. And we can talk about that later. So any questions that you do have, you can ask. But just as a quick rundown and summary, repressible operons are operating in anabolic pathways used for synthesizing end products. And if the end product is in high enough concentrations, then it is going to allosterically inhibit or attach to the repressor, which is going to turn off the expression of those genes. Inducible operons are going to be off in their normal state, and they usually are going to function in catabolic pathways to break down for digestion, things that might be in their environment. And if the nutrient is there, then they can act again to turn off the repressor protein. It will detach from the operator and will allow for transcription. I hope that helps. If you have questions, please let me know.